Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the living organism. And in this context, so far what we have discussed about the classification of the living organisms, and then we have also discussed about the evolution of these organisms. So and in the previous module we have also discussed about the we have discussed about the cells. So we have discussed about the prokaryotic cell or the eukaryotic cell. While we were discussing about the prokaryotic or eukaryotic cell, we could understand that the cellular functions are being performed by the tiny biomolecules which are present in these cells. And uh, so to understand the activity or to understand the cellular functions, it is important for us to understand the biomolecules. And in that context, in this particular module, we are discussing about the different types of biomolecules. And if you recall, in our previous lecture, we were discussing about the nucleic acids. So we have discussed about the DNA as well as the RNA, and we have discussed about the structure as well as the different functional properties of the biomolecules, the nucleic acids. And the nucleic acid is required for maintaining the genetic informations. So mostly the DNA is the major form of genetic genomic DNA, or it is the form which actually carries the genetic information from the one generation to the next generations. But where there are exceptions, where in some cases the RNA is also be the major molecule which is utilized for carrying the genetic informations. So in today's lecture, we are going to discuss about the another biomolecules, and then another biomolecule what we are going to start discussing about the carbohydrates. So let's start discussing about the carbohydrates. And so what we were discussing about the different types of biomolecules, and we have discussed about the DNA and RNA. And the DNA and RNA is required for storing as well as maintaining the genetic information from the one generation to the next generations. Except viruses, the DNA is the major molecule which is responsible for storing the genetic information in most of the organisms. Now in today's lecture, we are going to start discussing about the carbohydrate. So when we say about the carbohydrate is required for mainly for the energy productions. But in some cases, the carbohydrates are also being part of the building blocks. They are also responsible for modifying the some of the proteins and some of the another biomolecules. Like for example, the carbohydrates are important for generating the glycoproteins and all those kinds of molecules. And these glycoproteins have a significant role in the various types of the biological activities, like whether it is for the biological recognitions or other kinds of things. So let's start discussing about the carbohydrates. So carbohydrates are commonly being called as the sugar, right? And so carbohydrates, in contrast to the protein and the lipids, the carbohydrates are mainly being utilized to provide as a source of energy to run the biological activities. Plants are the primary production of the carbohydrates by the utilizing the atmospheric carbon dioxide, water and the sunlight. If you recall, we have discussed about this when we were discussing about the chlorophyll, right? Or when we were discussing about the chloroplast. So what we have discussed is that the carbohydrate, plants are actually sequestering the carbon dioxide from the atmosphere, and that's how they are mixing that with the water. And in the presence of the sunlight, uh, it is actually giving the sugar molecule. So it is actually giving the sugar molecule, and this is the sugar, right? And why it is required to the sunlight, it is required for running the light reaction, and it is sequestering the carbon as well as the water molecules to generate the simple sugar molecules. So carbohydrates are present in very simple as the monosaccharides to complex form as the polysaccharides as well as the glycoconjugates. So let's first start discussing about the monosaccharide, which is the simplest sugar, right? So this is the monosaccharide, commonly known as the glucose. So monosaccharides, it is the simplest carbohydrates, aldehyde or the ketone with two or more hydroxyl group. The backbone is made up of 3 to 6 carbon with the hydroxyl group attached to it, and the monosaccharides are always being named as per the number of carbon atoms are present. So these simple sugars are named by the number of carbon atom present in each molecule. For example, triose. Triose is the sugar which actually going to contain the 3 carbons such as the glyceraldehyde. So what you see here is the number of examples of the monosaccharides. So here you have the 1 carbon, here you have 2 carbon, here you have 3 carbon, 2 carbon, and here you have the 3 carbon, then you have the 3 carbon, you have 3 carbon, and so 1 carbon it is called as D-glycero. Then if it is a 2 carbon, then it is called as the erythro, and if it is 3 carbon, then there are many examples of 3 carbons like the arabino, lyxo and all that. So mostly it is being called as the triose. So if it is a, this commonly known as the triose, whereas if it is a 4 carbon, then it is actually going to called as tetrose, such as the erythrose, and then it also has arthriose or the threose. Then if it is a if it is a 5 carbon, then it is going to called as pentose, for example the ribose. And then if it is going to contain the 6 carbon, then it is going to called as the arabinose, xylose, lysose. And if it is 6 carbon, then it is going to called as hexose with the 6 carbons such as the glucose and fructose. So what you see here is that single carbon examples like the glycero or the 2 carbon examples such as the D-erythro or the D-threo, and the 3 carbon examples like the arabino, lyxo, ribo, and then it is also the 3 carbon examples. So I have given the many examples, and then it is the 4 examples, the 4 carbon examples where you have the D-allo and the D-galactose. So this is the 5 carbon, right? And so these are the simple monosaccharide sugars what is present, right? And so these carbon atoms, which are chiral centers to give the stereochemistry sugar found in the nature, right? So these, if you see a simple carbon sugar, what you see here is that all the carbon atoms are actually having the attached to the different types of groups, right? So this one, for example, it is on one side attached to the hydroxyl group, one side onto the OH H group, and the other side it is actually adding with the aldehyde group. So this is actually another first carbon chiral center, second chiral center, third chiral center, fourth chiral center. So this actually gives the stereochemistry which is found in the nature, and because of that the carbonyl carbon which is present onto the first carbon is actually being attacked onto the last hydroxyl group, and that's how it is actually going to give you the cyclized products. So it can give you the 2 different types of cyclized products. So the reaction between the first carbonyl group and the 5th carbon gives the cyclized form of the sugar. And that's how the sugar can actually exist in the 3 different forms. It can actually exist in the linear form, or it can actually be able to exist in the 2 different types of cyclized forms. Either it can be alpha form or it can be a beta form. Apart from that, we are going to discuss all other kinds of the, because it has a stereoisomeric carbons, so that's why it has different types, many different types of stereoisomers. As far as the cyclization is concerned, it can actually be able to form the 2 different types of cyclization products. The first product, which is called as the hemiacetyl or hemiketyl formations, that is occurring between the adduct formation between the carbon and the aldehyde groups. So the linkage formed between the aldehyde group and the alcohol is called as the hemiacetyl linkage, and this is present in the six membered pyranose rings. So for example, this is the glucose, right? So if the one or fifth, if the one is actually making a cyclation product with the 5, then it actually be able to form the cyclation product, right? So if it is, this is the aldehyde group, and if it is attached onto the fifth carbon, so that's how it is actually going to form the hemiacetyl formations, and that hemiacetyl form can be represented in two different forms. So what you see here is actually the Fischer formula, where the carbon is connected to this one, or it can be actually be able to form shown into the Haworth formula. So this is the Haworth formula, where the hydroxyl as well as the hydrogen groups are being placed onto a plane, and it is actually going to show you which one is going up on the top of the plane or which are on the bottom of the plane. So once you actually going to have the carbonyl group which is attacked onto the fifth carbon, and it will going to form the hemiacetyl groups or hemiacetyl linkage, that can be represented by the two formula, Fischer formula and as well as the Haworth formula. Same is true for the another one, right, where you can actually have the hemicatyl formations. So hemicatyl formation, the linkage between the keto group and the alcohol is called as the catyl linkage. But this is forming into the 5-membered ring such as the Finrose ring. So what will happen when you have the carbonyl group, or when you have the keto group, and that keto group is present onto the second carbon, so that keto group is actually going to attack onto the fifth carbon, and that's how it is actually going to form the hemicatyl formations, and that's how it is actually going to have the two different forms. This is the this is the Fischer's formula, and this is the Haworth formula. So same way, it's going to show you the 5-membered ring, and then it is going to show you the placement of the different groups either on top of the plane or the bottom of the planes. The cyclization is actually generates the additional chiral centers, and that contributes into the stereochemical complexity of the molecule. For example, this is the glucose molecule, where you have the carbonyl group which is on the number 1, then you have the number 2, 3, 4, 5 and 6. So all these, when there will be a cyclation product between this and this, that is actually going to form this cyclization product, right? So that's actually going to give you the additional chiral center, because initially this one, the first carbon, is actually not having any chirality, right? It is actually been attached to the oxygen on one side, hydrogen on one side. So this one is not this is a achiral molecule, but when there will be a cyclation product and this is going to connect it like this, then it is actually going to acquire the chirality. So because of that, it actually can have two OH on this side and H on that side. So that's how it is actually going to be represented like this. So now you see, this is additional chiral center what is going to be developed onto the glucose molecule, and because this can be represented in two different way, it can be either the OH onto the bottom side, or OH can be on the top side, and that's how it can actually be able to adopt either the alpha form or to the beta form. So depending on the rotation of the different types of groups or the ability of the sugar to rotate the plane polarized light, it can be either be represented by the, it can actually be able to have the dextrorotatory sugar or to the levorotatory sugars, and these dextrorotatory sugar is represented by a plus sign or to the small d sign, whereas the levorotatory sugar is actually going to be presented by the negative sign or to the small l sign. Apart from, because as I said, because it has the different types of chiral center, it can have acquire the ability to rotate the plane polarized light. It actually can have the different types of stereoisomers. Now, one of the stereoisomer is the enantiomers. So enantiomers are actually be the mirror image to each other. So if you keep a mirror here, right? If you keep the mirror here, what you will see in this one is actually going to be the mirror image of this. So you know that all the groups are going to be flipped to the 180 degrees. So for example, in this one, except the first group, all other groups, like see, the OH is on this side, on this one is OH is on this side, the OH is in this side, this is OH in this side. So that's why all these are actually being flipped by the 180 degree, and that's why this form is called as the L form, and this one is called as the D form. So what I say is, don't be get confused when you see the L and D versus the small and small d, right? This small L is actually the levorotatory, and if this is D is actually called as the dextrorotatory. Whereas here we are actually going to show you the enantiomers, where you have the capital L form or to the capital D forms. Then we have, so this is what he will explain here. So D sugar, so the compound that have the last asymmetrical carbon in the same orientation, that is called as the D forms, whereas the L form, it is related to the L glycosidase. So what you see here is, if the last asymmetrical carbon is actually having the groups onto the same side, right? This one, you see, this one has a functional group which is on this side, and this one also has a functional group onto this side, then this is called as the D form. Whereas in the L form, it is actually going to be in the reverse orientation. So the group is actually onto this side, whereas the last carbon has a group on this side. So they are actually being presented into the two different modes, and that's why these two are actually going to be the mirror image as well. So this is the D form, and this is the L form. Same is true for the glucose also. In the glucose, this is the last asymmetrical carbon, right? So if the last asymmetrical carbon, the groups are on this side, and if the this one also has a group on this side, then this is called as the D form. Whereas in the L form, it is actually going to be on the reverse orientations. Now we can have the another kind of isomerism, that is called as the epimers. So the isomers differ as a result of variation in the configuration of the OH and H on the carbon atoms as 2, 3 and the 4 glucose molecule. So you can see in a glucose molecule at 2, 3, 4 and 5 you have the hydroxyl groups, and that's why the positioning of the hydroxyl as well as the H group on the different carbon atoms, or the differences between the different orientation of the OH and H on the different carbon atom, you can have the different types of epimers. For example, this is the D-mannose, because you have the epimerization of the OH groups, right? So OH is on the this side, right? So OH is on the this side. So this is called as the epimer at carbon 2. So, and that is present in the D-mannose. Similarly, you can have the epimerization in D-glucose, so that here also you can have the epimerization at carbon 3. So you have the carbon 3 at different orientations. So this is the standard molecule where you have the epimerization, and this is going to be used. Then is in the case of D-galactose, you see the D-galactose at the carbon 4, the orientation of the OH is on this side. So this is actually going to be different from the other sugar. So for example, in this one you have the OH on this side, in this one also you have the OH on this side, and so in the glucose, what you see here is this is the standard molecule. So this is going to be used for comparison purposes, right? Then we have the anomers. So anomers are actually been orientation of the alpha, or anomers means alpha or beta forms. So anomers are the monosaccharide that differ in the configuration of the OH group onto the carbonyl carbon or to the anomeric carbon. So you can see that this is the linear chain D-glucose molecules, and when it is actually going to go through with the hemiacetyl linkage formation, it actually going to generate the additional chiral center onto this first carbon. So onto the first carbon, if the OH is onto the right side, right, then it is actually going to be called as alpha form. Whereas if the OH is going to be onto the left side, then it is actually going to be called as beta form, right? So this is the alpha D beta form, this is the beta D beta form, right? Then we have the mutar rotations. So monosaccharides containing the asymmetrical carbon atom rotate the plane polarized light. The change in the optical rotation when either form of the glucose is allowed to stand in a solution is the called as mutar rotation. So you can see here is this is the alpha form and this is the beta form, right? So in the alpha form, you see the optical rotation is 100 plus 112 degree. But if you allow this small sugar to stand on into the solutions, what will happen is it is going to return back to the linear form. So with the conversion of this particular sugar into a linear form, which is the D-glucose form, it is actually going to change its optical rotation. So it is going to have the optical rotation of plus 52.5 degrees, 5 degree. Whereas same is true for this one also. If you see the beta form has the optical rotation of plus 19 degree. But on standing the sugar for into the solution, it is actually going to get converted into the linear chain, and that's how the optical rotation is going to be changed to 52.5 degree. Then the sugars actually can come together and they can be able to form the disaccharides. So when you have the two different sugars, right, you can have the hemiacetyl formation, right? So on the both the sugars, so what will happen is that there will be a dehydration reaction. So there will be a dehydration reaction, and because of that there will be a loss of water molecule and there will be a linkage which is going to be formed between this and this. So what will happen is if there will be a loss of water and this is actually going to lose, then there will be a linkage which is going to be formed. This is going to be called as the glycosidic linkage. And the disaccharide what is going to be formed, and you can read this as alpha D glucopanosyl 1,4 D-galactosidase. Why it is called as 1,4? Because you are actually making a linkage between the first carbon and the fourth carbon. So the OH what is present onto the fourth carbon and the first carbon is involved into the formation of the glycosidic linkages. So the individual monosaccharides utilizes their terminal hydroxyl group to form a glycosidic linkage to form the disaccharide with the loss of water. With the addition of the water, it can be hydrolyzed to form the individual monosaccharides. Different monosaccharides can participate to give you the different types of sugars. Same is true like if you take the disaccharide, for example, this is the maltose, right? So if you take the maltose and if you add this water, then what will happen is the water is actually going to hydrolyze this particular bond, and that's how you are actually going to have the two sugar molecules. So you are going to have the alpha D glucose, see the OH is onto the lower side, and you are going to have the beta D glucose where the OH is onto the top side. Glycosidic linkages what is being formed into the sugar could be of two different types. You can have the O glycosidic linkages or to the N glycosidic linkages. O glycosidic linkages, when the hydroxyl group onto the anomeric carbon of a sugar react with a alcohol of the another sugar, then that is called as the glycosidic O-linked glycosidic linkages, such as just now we have seen here, right? In this one also we have seen, right? This is the O-linked glycosidic linkages, means it is actually going to be between the first and the fourth carbon. So this is the anomeric carbon, right, where you have the OH, right? So that's OH is actually making a condensation with the another OH onto the fourth position, and that's how it is actually going to form the O-linked glycosidic linkages. These kind of linkages are present in the disaccharides, oligosaccharides, and they are also present into the polysaccharide, which means the O-linked glycosidic linkages, glycosidic linkages are mostly being present into the sugar molecule. So sugar to sugar actually. Then we have the N-linked glycosidic linkages. So hydroxyl groups onto the anomeric carbon of the sugar reacts with an amine. They are present in the nucleotides, RNA and the DNA. So When you have the sugar and you want to see the N-like glycosidic bond formations, you can see that into the formation of the nucleotides. So remember that when we were discussing about the nucleotides, when we were discussing about the nucleic acid, right? So nucleotides are being formed when the sugar is actually making a linkage with the base, and I said, you know, this base linkage is actually going to be the glycosidic linkages. So this is actually going to be the N-linked glycosidic linkages. So that is actually going to give you the nucleoside, and when it is actually going to make a pair with the phosphate, then it is actually going to give you the nucleotides. So that is what is called as the N-linked glycosylations, N-linked glycosidic bond formations, and that is present in the different types of nucleotides which are present in the RNA or the DNA. Then type of glycosidic bond or linkages. So you can have the two different types of bonds. You can have the alpha bonds, right? Alpha 1,4 linkages, just now we have seen, right? Or you can have the beta 1,4 linkages, right? So if the anomeric hydroxyl is below the hydrogen, right? So if the anomeric, then it is actually going to be called as alpha linkage, alpha glycosidic linkages. So it still be O-linked glycosylation, O-linked glycosidic bond formation, but it is going to be called as alpha linkage, which means it is going to be called as alpha 1,4 linkage. So for example, in this case, this is the maltose, right? So what we have is we have the OH onto the lower side, right? So this is the alpha form, right? So then if it is in reacting with the another OH onto the fourth position, so this is the number 1 position, which is the anomeric carbon, right? And this is the fourth position. Then it is actually going to be called as alpha D glycopyranosyl 1,4 alpha D pyranoside, right? So this is actually going to be a alpha 1,4 linkage, right? And why it is called as alpha 4? Because the OH what is being involved into the formation of the glycosidic linkage is actually be present into the alpha conformations. Then we have the another kind of thing where you have the lactose, right? So here the OH is present in the beta conformation. So if the what is the beta form bond? If the anomeric hydroxyl is above to the hydrogen. So if the hydroxyl group is present into the beta conformation or beta form, and if it is involved into the glycosidic bond formation, then that will be called as beta linkage. And for example, here we have the beta 1,4 linkage. So here what we have is beta D galactopyranosyl 1,4 beta D galactopyranosyl. So here this is the structure of the lactose. Same kind of glycosidic bond are actually going to be formed to give you rise to the polysaccharides. So these are the polysaccharides. So here the condensation of the different polysaccharides or the disaccharide will give rise to the polysaccharides. Polysaccharides are the storage form of sugar, and it gives hydrolyze to give the monosaccharides to participate into the metabolism to produce the reducing equivalent to provide the energy via the electron transport chain. The examples are like the starch, which is the storage polysaccharides present into the plants, or we have the glycogen, which is a storage polysaccharide what is present into the animal. Both the starch or the glycogen are actually being stored into the food as in the form of a food, and that's why they are actually going to be used for the energy production. So what will happen is that polysaccharide, whether it is a glycogen, for example, right? So glycogen is actually, whenever there is a requirement of the energy, the glycogen is actually going to be converted into the glucose, and then the glucose will go into the metabolism and it is actually going to produce the energy. Same is true for the starch also. As the anomeric carbon participate in the formation of glycosidic bond, it has lost its ability to form the linear form of sugar. As a result, it becomes a non-reducing sugar. In the disaccharides or the polysaccharides, the terminal sugar has the free anomeric carbon and called as the reducing sugar. So if I want to know whether a particular sugar is, so you can have the two different types of sugar, right? If you can have the sugar which is reducing sugar, right? So or you can have the sugar which is non-reducing sugar. So what is mean by the reducing sugar? So reducing sugar is in the case of disaccharides, right, where the terminal sugar has the free anomeric carbon and called as the reducing sugar, whereas in the case of polysaccharide, as the anomeric carbon is also participate into the glycosidic bond, it has lost its ability to form the linear sugar. As a result, it becomes a non-reducing sugar. So let me explain this. For example, in the case of disaccharide, what will happen is, like suppose this is the one of the disaccharide sugar, what will happen is that this is going to be formed, right? So this is going to be formed like this, right? But this is your anomeric carbon, right, where you have the OH, and this is free. So this, for example, if this is the maltose, which is so I'm not showing the all other bonds, but so this is the alpha 1,4 glycosidic linkage, and that's how it is actually going to be. And this OH is free, so this actually can participate into the reduction and oxidation reactions, right? So this is going to be called as the reducing sugars. Whereas in the case of polysaccharide, what will happen is that you can imagine that this OH, if I convert the maltose into a polysaccharide, or if you add some more sugar, right? So what will happen is this OH is again going to form an bond. So if you see the polysaccharides, like it's like this. So it's like this, right? So this is the anomeric carbon, right? So this is your anomeric carbon, this is the 1,4, right? So if it is 1,4 linkage, this one is actually the fourth linkage, this is the 1, this is 4, this is 1, this is 4. So you see the anomeric carbon is actually being involved into the formation of this particular bond. So there is no free OH at the anomeric carbon what is present. So that's why this is actually going to be called as the non-reducing sugar. Now the question is how you actually can be able to know whether the given sugar is actually going to be the reducing or to the non-reducing. To know that, you can actually be able to test with the ability of a sugar to reduce the given compounds. So glucose and the other sugars are capable of the reducing ferric or the cupric ion in the solution and are called as the reducing sugars. They reduce and the color Benedict and the Fehling solution with the aldehyde groups onto the sugar. So what is the Benedict test? So if you want to know whether the sugar is reducing or non-reducing, what you can do is you can do a Benedict test. So Benedict test is actually being a very simple test where what you can do is you can take the Benedict reagents. So what is Benedict reagents? Benedict reagent is a combination of the anhydrous sodium carbonate, sodium citrate, and it has a copper 2 sulfate pentahydrate, which means it is actually going to have the copper 2 plus, right? And and this is going to be a blue colored reagent. So if you don't have a sugar, it is going to give you the blue colored reagent. So what you have to do is take a clean test tube and place inside the test tube 1 ml of the solutions. Okay, so whatever the sugar solution you want to test whether it is reducing or non-reducing, you take the 1 ml of that particular solution. Then you put the 10 drops of the Benedict reagent into the test tube. Bring the solution to heat in a boiling water bath for approximately 5 minutes. So what you can do is you can take this 1 ml solution into the test tube, and then you add with the help of the right. So you add the sugar solution, and then you add the small amount of the Benedict reagents, and then you are actually going to keep this into a water bath. Okay, so when you boil this in a water bath, there will be a reaction between the sugar and the copper that is present into the Benedict reagents, and as a result you are actually going to see the change in the color into the particular Benedict reagents solution. So what you will see is that if there will be no sugar what is present, or if the sugar is non-reducing, then it is actually going to give you the no change in the color. So if there will be no change in color, that could be because the sugar is non-reducing. Then if it is a going to give you a slight green color solutions, then that will be called as that it is has a very small amount of reducing sugar. Then it is actually going to give you, if it is going to give you the yellow color, then it is going to be a moderate uh, amount of the reducing sugar. And if it is give you the red color, then it is actually going to say that you have a large quantity of the reducing sugar. Apart from the Benedict reagents, you can also use the Fehling solutions, and that also is going to give you the similar kinds of results. So with this, we have understood the different types of properties of the monosaccharides. We have understood the structure of the carbohydrates, and we have also understood how you can be able to distinguish between the reducing and the non-reducing sugars. Apart from that, in this particular lecture, we have also discussed about the different types of isomerism, what is found into the sugar solution, sugar monosaccharides, and how the monosaccharides are coming together to give you the disaccharides. It is actually forming the glycosidic linkages, and these glycosidic linkages could be the alpha type or to the beta types. And apart from that, we can have the two different types of glycosidic linkages. Either you can have the O-linked glycosidic linkages or to the N-linked glycosidic linkages. So with this brief discussion about the structure as well as the properties of the carbohydrates, I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss about the metabolism of the carbohydrates and how the carbohydrates are participating into the energy productions. So with this, I would like to conclude my lecture here. Thank you.